that we covered. We finished, last time when we met, we finished chapter 2, the chemistry chapter, right? So we're going to start a new chapter, chapter 4. And chapter 4 is called Functional Anatomy of the Prokaryotic Cell and the Eukaryotic Cell. And the focus for this chapter is going to be on the prokaryotic cell, right? Eukaryotic cell, I think maybe like the last 9 slides or so are eukaryotic cells, but that's self-study. Again, it's indicated on the lecture schedule. There's an asterisk that says parts of this chapter is self-study. It's the eukaryotic stuff that's self-study. But again, I just go through it real quick. Eukaryotic. The reason why it's self-study, you guys actually probably know a lot about eukaryotic cells, right? Because eukaryotic cells include the kingdom that we belong to, which is animalia, right? So animal cells are eukaryotes. You've taken anatomy, physiology, all of that focuses on the eukaryotic cell. This course, micro, is probably the only one that's dedicated mainly to the prokaryotic cell. Because the answer is right here. I was going to ask you, what's a prokaryotic cell? The typical example I told you is bacteria. These terms are used interchangeably, bacteria and prokaryote. They're one and the same. A bacteria is a prokaryote. A prokaryote is a bacteria. By the way, what kingdom is this? Monera. Right, this is kingdom Monera. We've covered this already. All right, and yes, the bulk of this course, I would say probably like 90% of this course is dedicated to prokaryotes. Not 100%, because you saw last week we covered in lab kingdom Protista, and this week, if you look at the schedule, we're covering kingdom fungi, which are not prokaryotes, those are eukaryotes. That's why I can't say it's 100% prokaryotic. This class, it's 90%, most of it is on bacteria for this course traditionally, all right? So yeah, the eukaryotic cell is going to be more of a self-study, all right? So let's start here with our, really our first official slide here. And we're gonna talk about the different parts or anatomy of, again, a prokaryotic cell. That's what we're focusing on for this first part of this PowerPoint. And again, what's the typical example of a prokaryotic cell again? Bacteria. So basically, I'm going to be talking about the parts of the bacteria in a moment. But we're going to talk about like size and things like that first. We're going to talk a little bit about the average size, and this is the average size, typically of something that's kind of maybe elongated, sort of bacteria. Elongated, it's like 2 by 8. But the units of measurements that we use are this thing called micron, or you can call this also micrometer. Micron typically is what we use, and it's this fancy U. I think it's a Greek letter. You don't have to know that. It's called mu, like mu-m. That's the symbol. And mu-m, I think we covered this in lab. The micron is a lot smaller than like millimeter, and certainly smaller than centimeter, because bacteria are very, very small. So we use this unit of measurement. And again, why do we need to know the average size? How is that helpful if we know the average size or the size period? The baseball team is having too much fun. It's bothering. Okay. Why is that important to know the average size or the size of the bacterium? Because you are going to eventually have to measure bacteria and record that information. Why do we want to record? It helps in identification. Okay, so let's carry this further. We've talked about this before, identification. So how does that help us if we know the name of the bacterium? If we identify it, it helps us to do what? Diagnose, and if you can diagnose, you can treat that effect. That's why it's important. Size is very, very important. Okay, and we'll get to do that a little bit. That's what we did the whole thing with the calibrating the micrometer. Remember we were talking about like the small divisions, how much each one was worth. And that was also, we can measure finding things. Okay, but please don't memorize these numbers. I'm not going to ask you the dimension. The take home is that we use to measure bacteria, we use microns, because bacteria are very, very small. That's basically what I want you to know. Don't worry about these numbers. That's not important here. Okay, these are 3 images here of visual field when you're looking at a microscope. Okay, and when you look at it, you kind of have, to describe what you're seeing, what you're looking at there in terms of the shapes that you see in your visual field. And we can describe that visual field as showing like monomorphic shapes or appearance. I should say appearance. These are more like appearance. the bacteria can exist as a monomorphic type of appearance or pleomorphic. Mono obviously means one, and pleo sort of means many or several. So these first two here, the purple and the yellow bacteria here, these are what we describe monomorphic. Sorry, monomorphic. First two are monomorphic. And what monomorphic, I told you, yes, mono means one, but what I mean by one is that one shape predominates. That's what I mean by one. Huh? Morphic, yeah, morphology, shape, structure, absolutely. So we have basically, like it's saying, one shape. Very good, you're breaking up the word. Morphic does mean shape or structure, right? So as you can see here, the first two I told you are monomorphic. So here, they are pretty much all or mostly spherical, aren't they? The purple ones. If you look at that visual field, they're mostly are all pretty much spherical. So that one shape predominates, the spherical shape. And over here, they're pretty much all are mostly rod shaped. There's no really variety here. One shape predominates in each of these visual fields. So these are monomorphic. The third one is a little different. This one is now pleomorphic. So because here we have several shapes present or exist. Pleo means several. And you do. If you look at this, there's variety here. They're kind of all elongated, but they're different looking, right? This one's kind of like really short. It's like an oval. And this one is a thinner and it's longer. And this one is long too, but it's thicker, right? So they're not uniformly in shape, right? They have a general kind of shape that they have in common, but you can't say that they're all you can't say that one shape predominates, right? So over here we have, like I said, several shapes. Several shapes present. And when you have several shapes present, it can mean one of two things, right? So why? Because of the two following. It could be that you have, for example, different bacteria. These are different bacteria. Maybe you have, I don't know, maybe I have 6 different bacteria. That's why they have 6 different shapes. Over here for the monomorphic, we have to assume it's the same bacteria. That's why they all look alike. Okay, they're all like clones of each other. But here, it could be that you have different bacteria. Or it could be that your culture got contaminated. Maybe you only grew one, you were trying to grow one type of bacteria only, and at the end it looks like this, so it could be contamination. You know, unintentionally you have several different bacteria in there, not on purpose. But number one was because you purposely did it. Number two is because it accidentally happened, because it got contaminated. All right. So there's a difference between monomorphic and pleomorphic. All right. So still talking about shape of, again, bacterial cells or prokaryotic cells. Right. Again, they mean the same thing. And there's 3 basic shapes here for bacteria. And this is going to overlap a lot in lab. So when you see in lab, I have a handout that I'm going to give you. It's also on Canvas. It's going to be repeated again. And that's okay, because repetition is good. Okay? Coccus means that they are round or spherical. I can't spell spherical. Spherical. Okay, round or spherical shape. Bacillus means rod shape. And under the heading of bacillus, there's this other one called coccobacillus. It still has the name bacillus in it. So it's technically still a rod shape, but it's kind of roundish, like a roundish rod, or what I call an oval. Have you seen an oval? I'll do my best attempt. It's not going to be good though. Let's listen back, better than my other spot. It's kind of rod, but it's kind of round, right? It's oval. Well, you know what I mean by oval. Not a perfect circle. It's just a little long, like an egg shape kind of thing, right? Yeah. And it's still considered rod though, even though, you know, it's kind of rounded, so I call this like oval shape. And our third shape is the spiral shape, which is gonna be kind of like, what's the best way to, you know, like coiled / curve kind of shape. And these are the three specific spiral ones that we have: vibrio, spirilla, and spirochete. All right, and all of these, what they have in common is motility. They are able to move. These bacteria, motility, remember, means movement. All three of these bacteria that are vibrio, spirillum, and spirochete shape, or are considered spirochetes, they're all spiral and they all have the ability to move. These other ones don't necessarily move. Okay, they don't necessarily. We're going to talk about movement. Again, we talked a little bit about movement, I think, when we covered Lepisa. I talked to you about flagella, but anyhow, we'll talk about flagella today too. All right, so that's what they have in common. And up here, from these pictures from your from your book, figure 4.4, right? These are referring to the spiral shaped bacteria. This one up here, this is the vibrio one. The vibrio is kind of described as a curved shape, or almost like a comma. You know, like a comma, like the punctuation mark. It's kind of bent a little bit, right? It's kind of like that. It's bent. So that's vibrio. This one B over here, this is your spirillum. All right, and this one down here is spirochete. Okay, and on the next slide, I'm going to compare and contrast the spirillum and the spirochete because they're slightly different. You can tell by looking at them, they're slightly different. One is kind of thick and one is thin. First of all, you can tell by this, but again, they're all kind of bent or kind of coiled or curved, right? All right, so let's compare and contrast the spirilla and the spirochetes, right? Okay. And again, of bacteria, we're all talking about bacteria, right? I'm gonna put next to spirillum, because they mean the same thing. You know, the Latin is gonna drive you crazy again. You don't need to know this. I'm not gonna test you. Spirilla is plural, spirillum is singular. Remember I told you about like bacteria, bacterium. It's the same thing. So when you see it, because they're like, probably saw this as spirilla, you're like, wait a minute, what's spirilla? You called it spirillum over here, now it's called spirilla. It's because I just made it the word plural. It's the same thing. All right. So let's compare and contrast these two in terms of characteristics. So the spirilla, right, this one down here, it's more of a rigid type of cell, and they're short, thick, curved, you know, coiled type of cells. And that's the opposite here. Spirochetes are more flexible, and they're just like skinny. They're just long and thin cells, right? So they purposely drew this one thicker to show you that they are thicker and short. Okay. In terms of how much coil they have in them, the spirilla is described as loosely coiled, but the spirochete is more of a typing coil type of cell. And I told you they all have the ability to move. The structures that they use to move with are going to be different. For spirilla, they're going to use an external structure called a flagella, and this is an external structure. Whereas on the spirochete, they're going to use the opposite. It's going to be now not an external structure, but it's going to be internal. Because what does endo mean to you? Inside. Same thing as internal, right? So this is an internal structure. Right? Here you can see the flagella. This is the flagella. Or flagellum. Singular flagellum. Right here on this one. Right? And again, this one is going to be this structure that it used for motility, for movement. You really can't see it. It's more on the inside. I have another slide for this one, but you can't see it. It's not going to be, it's not external. So it's very different. But again, they cause movement, right? Because again, that's what the spiral shape bacteria can do. Yes, question?
Compared to the spirilla and the
spirochetes.
Spirochete, which one's more common for bacteria to have the shape?
Which shape is more common?
Yes.
That's a good question. I want to say it's the spirochete because a lot of the diseases we covered in this class are going to be like spirochete type of diseases. And talk about syphilis, Lyme disease, things like that. Those are all called spirochete diseases. So, as far as like pathogenic, the ones that cause a lot of disease, I would say it's the spirochetes. Yeah. Okay. Yes, question.
Do all Spirilla have flagella?
Do all Spirilla have flagella? I think so, because they have to be able to move. They have to have some sort of structure.
So if they wiggle like that, they can move.
Yes. There's a structure, there's some sort of appendage or structure that allows them to move, right? They have to have something, just like when we cover, I know this is not Protista, but in lab, we cover the cilia, pseudopodia, flagella, the adhesins. They have to have some sort of structure that allows them to move, and it's the flagella. I think later on I'm gonna talk about the flagellum can be in any location, it doesn't have to look like this. I can have another one on this end, or it can be like all over.
So how do you help in Fimbria and flagella?
Fimbria are going to be, I have a slide on that, Fimbria I think are a lot shorter. They resemble almost like hair-like type of structures, they're much, much shorter. And their arrangements, they're gonna be like all over. Flagella you can, but, I mean, I have another slide later, but you have, if this is my cell, I can have a flagella here, or they can be like this, look like a ponytail, there are several, but they're all on one end. Or, again, I can have it this way, or whatever. Okay? All right, sure. Let's talk about arrangements. And by arrangements, we're talking about, how do the cells exist? Do they exist in pairs? Well, first of all, do they exist as single cells? In pairs, we're asking all these, what's what we want to find out? In pairs or in chains, in clusters, all of these things, that's what we mean by arrangement. How do they exist? How do they exist as? Right, like your typical bacterial cells. They can exist in pairs, so if you see them in pairs, you use the prefix diplo, right? Di means two, right? So you kind of put it together with the shape, because this is referring to it like the shape, right? The prefix diplo we know means two. So if something we describe it as a diplococci or diplococcus, how would that look? Would you be able to draw that out? Diplo means again what? Two, and cocci means what? Round, spherical. That's diplococci. Right? How about diplobacilli? Two what? Spheres? No, two rods. And the rods have to be written actually this way, end to end. And it's two of them. You know what I mean by end to end, like tail to tail, whatever, head to tail, whatever, end to end. Diplobacillus is not this one. I'll show you another one. People are like, oh, well, isn't that, what's the difference? This is two also, and they're bacilli, right? True, but Diplobacillus is only this one. It is not this one. I'll save this one for lab. It has a different name when they're side to side. So, again, not side to side. These are end to end for the Diplobacilli. This is another name, which I don't mix that with lecture. I'll save that for lab on Thursday. You'll get what it's called when they're side to side, but it's not Diplobacilli. Clusters, maybe they exist in clusters this time, and so now you can't use, diplo, now you use the prefix staphylo. Staphylo means clusters. So staphylococci would be what? A cluster of circles, of spheres, right? Whatever, a cluster, that's staphylococci, the name gives it away. The prefix strepto is referring to chains, so streptococci is a chain of several, more than two, otherwise it'd be called diplo, but it's a chain of spheres, of circular bacteria, right? How about streptobacilli? A chain of bacilli, but again, end to end. It's not this. People are like, well, how come I can't do this? That's called something else. I will save that for lab. That's not that. Okay, the chain has to be this way, end to end, not sideways, not side to side. Okay, that's streptobacilli. These last two, the tetrad and the Sarciniae, and again, we covered these in lab again, so there'll be a lot of overlap with the lab material this week. These are only for cocci. Certain cocci can be described as tetrads, certain cocci can be described as Sarciniae. The tetrad is like a group of 4. But they're arranged that way, like in a nice little like square kind of thing. They're 4. Okay? The Sarciniae is similar, but it's doubled. Instead of a group of 4, you have a group of 8 because they are stacked. 2 tetrads stacked, we'll call it that way. 2 tetrads stacked. So I don't know if I can draw this one really well. So here's the first layer. It's like 2 layers, right? And then I have maybe another layer stacked directly on top of it. It's gonna be hard for me to draw, but I'll do my best attempt. It's like you have another one on top of it. It's like a 2-layer thing, right? 2 floors. Can you get it? Huh?
Like an overlap?
It's directly on top, like a cake. I have the first layer, and then I put another second layer right on top. The first layer has 4, another layer on top that has 4. It's like a 2-dimensional kind of thing.
For the cluster, is that one also Priscilla or is that one?
This cluster is a very good question. The clusters is only for cocci. This cluster here, yeah, only for cocci. Good point. There is no such thing. If you're wondering what happened to Staphylococcus, that doesn't exist. There's some analogous thing. I'll save that for lab, but we never ever call anything Staphylococcus. It doesn't exist. Yes.
For the cell arrangement for prokaryotic cells, will we most likely see the cells in pairs or clusters or
It varies. You can see some of them, they're single, some are gonna be in clusters, so you'll get to do that this week in lab. Okay, but we pay attention to the one, I'm not gonna ask you like, what's the arrangement, because it's not one answer. I'm gonna ask you what's the most common arrangement, maybe what's the second most common arrangement.
Yeah, that's what I'm asking.
Yeah, you won't have just one, usually, typically. Alright? I told you each exam has about 2 to 3 diagrams. Here's one of your diagrams on your exam. I'm already telling you what the diagram. This is one of them. This one, however, is completely labeled for you. I have another one on Canvas. This is on Canvas already if you go to the Unit 1 module. I have another one where it's blank if you want to practice with. I'll probably pick like maybe 2 or 3 things that I want you to identify. You just don't know which one you're going to pick. So you probably have to say them all, but I'm not going to have you label the entire thing, picture though. But I mean, you still have to know it all, right? Okay, so, yeah. So we're going to talk about, in the next couple of slides, we're going to talk about, again, each part that is present in a bacterial cell, right? Prokaryotic cell. We're going to talk about the location of that structure, what is it made out of, what is its function, things like that, right? In some sort of logical order. I want to start from the outside and work my way inside to the nucleus, right? So, and we'll be covering most of these, not every single one, but most of these, I think I lied. I think pretty much all of them that I am going to cover. All of these on here, okay? And there should be some logical order to kind of how you organize the material, because I'm going to organize it basically by bullet points and so forth. But I recommend doing this material, putting it into a table. It's much better organized if you do it into a table. This table is already open. It's on Canvas for you in the miscellaneous handout folder. And it goes in the order how I'm covering things. So I'm going to talk about, shortly after this slide here, I'm going to talk about the glycocalyx. So you should know the location, that's the first column, the location of it, or maybe a description of the glycocalyx, like, you know, kind of what it is. Here, composition, what's the glycocalyx made out of? So anyway, composition, what's the function of that glycocalyx, right? And then, you know, I don't know, whatever. I always like to have a miscellaneous column, you know, or you can put examples. Here, whatever I don't know where it goes in my miscellaneous box, right? And then after that, I'm gonna cover flagella, the axial filament, etc., going in this order. So probably today, I'll probably finish somewhere, hopefully, in the cell wall, and then we'll have to continue this next time on Thursday, and then I have to hopefully finish the rest of it during the 20 minutes that I'll take from last time. It's a lot of material in this chapter. And Then after inclusion bodies down here, then the slides begin for the eukaryotic cell, which is like the self-study part. Okay, so some sort of order to this, and then again, you can change this table if you want, or you can add another column, or whatever you need to do. And again, you're not turning this into me, this is just to help you organize it, so you can do it in groups, if you have a study group, or do it on your own, or maybe Angie will do it during SI, or you can ask her as well, something like that. Okay, so let's start in order with top here with glycocalyx. Okay, and I'll probably point out in the beginning, like, okay, this is the description, this is the function, things like that, just to kind of get you oriented, all right? But the glycocalyx is secreted by bacteria. By some bacteria, not all of them can do this, it's secreted by some bacteria. So that's what I mean by this is kind of optional. It's not always present. Sometimes it's present, sometimes the glycocalyx is not present. It's just, it depends on the bacteria. Some can secrete it, some do not. Okay, so maybe I would put this in the miscellaneous box, right? Because this is not necessarily the composition. I can tell you, this has nothing to do with composition or location, right? So you kind of need to put things in the correct box, right? So this is kind of more miscellaneous information. So again, glycocalyx is optional, not always present. Now here is the location, right? Location, where do you find a glycocalyx? It's going to be outside of the cell wall, external to the cell wall, right? So if this is the cell wall, let's say this is my bacteria, right? And here in red, I have, this is going to be the cell wall, CW, for the cell wall. And if it does secrete or produce a glycocalyx, the glycocalyx is going to be external or outside of the cell wall. So this green here represents the boundary of the glycocalyx. But in actuality, it's really all of this. I left it white, but all of this. I won't do it all the way, but you know what I mean. All around the whole cell, all of this surrounding the entire cell. This is your glycocalyx. Right, because when I used to just do the green thing, people would think, oh, it's just the green thing. No, I mean, it's the entire thing, this entire space here is the glycocalyx. It surrounds the cell wall of the bacteria. Okay, description of it. What's the description of glycocalyx? The description, it's basically like a gelatinous type of material. It's not liquid per se, but it's kind of gelatinous, and they describe it as being viscous. Do you know what viscous means? What is viscous? Huh? Thick, right? Kind of like syrup, like maple syrup. Syrup is not water. I don't pour the thing that's just going to be water. It's kind of like a thick, or like motor oil. You probably have no idea because you've never changed it. How many people change their own oil? You change your own oil? Oh my goodness, you want to change mine? No. All right, I used to do that when I was in high school. I like that because it's not worth my time anymore. But if you ever poured motor oil out, same thing. It's kind of takes its time. Kind of like syrup, right? Viscous. Not completely liquid, okay? So that's the description. Composition-wise, what is a glycocalyx made out of? So this is composition. It can be made out of either a sugar, right? Because you know polysaccharides are carbohydrates, right? Which are sugars. So it's made out of sugar, or it can be also made out of polypeptide. What does this mean to you when I say polypeptide? Protein, right? Because remember, it's peptide bond, we talked about that. So yes, basically the glycocalyx can be a protein glycocalyx or a sugar, carbohydrate. Carbohydrate glycocalyx. Okay, that's, if you're doing the chart, that will maybe go into the composition box. And there are two types of glycocalyces, right? And by the way, don't get, again, more Latin. Glycocalyx is singular, glycocalyces is plural. Okay, and it's a Latin thing. Just like in anatomy, what was it? What did we say the other day? It was the calyces, right? The major and minor, the calyx and the calyces, same thing. Okay, so don't get confused with, oh my God, what's a calyces? It's the same thing. Two types. There's the capsule glycocalyx or the slime layer glycocalyx. Right? They're different in terms of what they're composed of, the composition, right? For capsules, these are carbohydrates. They're sugar composition. They're made out of carbohydrates. So by default, slime layer is made out of protein, so protein composition. Going back to capsules, capsules are pretty thick, like I'm showing here. They surround bacteria, surround the cell wall, and they're pretty thick, and they firmly attach to the cell wall. They're not coming off. They're pretty much attached to the cell wall. Whereas the slime layer is thinner, it's unorganized, and it's just very loose. It can come off real easily. Right? So here's some illustrations that I found from the internet, right? And again, this white space here, that is your capsule. And remember, the capsule is made out of a carbohydrate, a sugar, and the slime layer is this other one, this other white area here. And again, this one is made out of a protein. Right? When you look at at capsules, right? The capsule glycocalyx under the microscope, it's going to have sort of this appearance here. We'll do a lab in a couple of weeks where the clear area is your capsule, or glycocalyx capsule. Glycocalyx capsule. It's basically the clear area. The dark thing in the middle, that's actually your bacterium. Right? And basically it looks like this. The dark area in the center, that's your bacteria. This is your glycocalyx capsule, this white area though. Okay. We'll have a lab on that, you'll be able to see the nice, clear, big capsules, clear area. That's actually one of your, that's the second demo quiz that you do. You have to prepare a slide to show me a capsule. If you can't show me a capsule, then you have to take a 0 on it. Because we have 2 demonstration lab quizzes, right? Remember, one of them was the gram staining. This is called capsule staining. We start plenty of time. We'll talk about this more in lab in a couple weeks about capsules. But you'll have a preview. So in this class, again, I know there's 2 types of glycocalyxes. We have the glycocalyx capsule, glycocalyx slime layer. I'm going to focus on the capsule for this course. We're not really going to talk much about slime layer because they don't really do much. So I'm going to focus the rest of the discussion of glycocalyx on the capsule. We'll talk a little bit more about it. Again, we know it's thicker, it's going to firmly attach to the cell wall, and it has 2 functions. So we're talking about functions. FXN functions. Here's the first one. The function of glycocalyx is to attach to host tissue. Very important if that bacteria wants to cause an infection, you have to attach to tissue. Otherwise, you don't establish an infection, right? So they have to attach to host tissue, and capsules are really good for attaching to host tissue because they are sticky. The capsules are sticky, so it makes it real easy to attach, right? Why do you think they're sticky? Exactly, because made of sugar or carbohydrates. Right? Have you ever touched sugar? It's kind of sticky after a while, right? So capsules are kind of sticky. That's great for them. Helps them attach to host tissue to cause infection. Second function of the capsule, it allows them to escape phagocytosis. Do you remember what phagocytosis is? Engulfment. It helps the bacteria escape engulfment, okay, phagocytosis, right? How does it escape? Because there's immune cells that want to engulf them, to destroy them. So how does a capsule help, for that so they won't get engulfed? It makes the bacteria look a lot bigger than what it is. I told you, we can have 2 bacteria. Here's and the same one, clones of each other. So I have this one here, and I have the other one here. You agree with me, same size and everything, but the one on the right is going to have a capsule. The one on the left does not have a capsule, okay? So when our immune cells, when we talk about this during exam 3, right, an immune cell, some sort of, white blood cell or whatever, is gonna want to engulf things, right? This is my immune cell. I'll put a little nucleus in here. is gonna try to engulf, and this one it can easily do, right? The same one, I don't know if I can redraw, but try to copy what I just drew. Oh my God, well, pretend it's the same one. Okay. Can't do it. It makes, this bacteria is protected thanks to the capsule, because the capsule makes it look a lot bigger than what it really is, right? Like Pac-Man, yeah.
Yeah,
It
it
makes
looks
it,
like a fucking Pac-Man.
right, it makes it look, the capsule makes it look a lot bigger than what it really is, right? I know I'm not that skinny, but if you put your arms around me, you can probably tackle me and knock me to the ground, but if I wear like the Incredible Hulk suit, you can't do that, right? It makes me look a lot bigger than what I am, and you're gonna have trouble knocking me down. It's the same thing what capsules do. Okay, that's a great advantage for bacteria. It makes them very difficult to get rid of, because they can attach to things, because they're sticky by nature, their composition, and they make the bacteria look a lot bigger than it is. So which one of these two bacteria is more dangerous then? Left or right? The right one, for these two reasons that we just said. Okay, here's some live examples. There's many of them. I just gave you two random examples of bacteria that have capsules. That's what we mean by encapsulated. That means that they have capsules. So a bacteria called Streptococcus mutans and Streptococcus pneumoniae. Let's look at this first one here. Let's reduce some old material. What's the genus name of this first one here? What is it, the genus name? Streptococcus, right? It's the first name. Don't we name things by genus followed by the what? Species. So mutans is the species, right? Capital S for genus, lowercase m for the species. So Streptococcus mutans. What's the shape of this bacteria? We just learned this. It's round because it's coccus. And what's the arrangement? It's a chain, because strepto means chain. So this is round, this is chain, we just covered that, right? So round chains, doesn't Streptococcus mutans look like this? Right, it does. Okay, the name gives it away. All right. Oh yeah, and Streptococcus mutans, this species of Streptococcus, this is found on teeth. Slash mouth, whatever, it's in the oral cavity, it's in the mouth, but particularly on teeth. This is responsible for cavities, the Streptococcus mutans. Alright, and then the Streptococcus pneumoniae, gee, I wonder what this causes. Pneumonia, the species name gives it away, this is what's responsible for pneumonia. Okay, and so yeah, the bacteria that causes pneumonia has capsules, and these are like huge capsules. This is some of the largest capsules out there. Right here, I drew a large one with a huge capsule. But again, just because they have a capsule doesn't mean that they look all like this. I can have another one here, still a capsule, right? But it's not the same size. The one on the red is a huge capsule, much bigger than the purple, right? But I'm just telling you. With Streptococcus pneumoniae, these bacteria that cause pneumonia, they have the reputation of having really huge capsules, right? Here's the image here, right? The clear area here, again, I should use purple. The clear area here surrounding the bacterium, that is your capsule. So let's take this one. I like this one, so let's just do that one. That is your capsule, right? It's the clear area. So what do you think that purple thing inside the capsule must be? That's your bacterium. Okay, so all of these have capsules around them. Okay, question for you, is this a slide of somebody that has pneumonia? This is the specimen that causes pneumonia, right? So, what do you think? Is this pneumonia? Are these, first of all, are these cocci? They look like cocci to you? No. And the one that causes pneumonia, is it a cocci? Well, round is cocci, right? Okay, so the answer was it. So the one that causes pneumonia, they are cocci. These are not cocci. To me, they look like they're what? Rod shaped. So no, this is not pneumonia. It can't be. Because again, Streptococcus pneumonia, look at the genus name. It's telling you the shape. These are cocci. These here, I'm asking you, these are not cocci, these are bacilli, so this cannot be pneumonia. Right? Okay. And also this reminds me for for some bacteria, the arrangement and the shape gives you the genus name. For some bacteria, the arrangement and shape equals genus name. Because you just told me the first word, Streptococcus, for both these bacteria, is the genus name, correct? And again, here the strepto part is the arrangement part. Arrangement, and the coccus part, the second part, is the shape part, correct? I just told you, arrangement, number one, followed by shape, gives me, for some bacteria, the genus name. Okay. All right. Any questions before we move on to the next structure? Okay, so we're done with the glycocalyx, the capsule. And we'll talk more about the lab in a couple weeks. Let's talk about flagella next, right? And again, this is plural. Flagellum just means singular, whatever. Flagellum, flagellum. Okay. Some miscellaneous again may or may not be present. It isn't always present. Some bacteria have them, some that don't, right? And if you have it, it's because it's for motility, for movement, right? I'll get to there when we get to function, which is the next slide. The location, right? It's external to the cell. We just talked about it. Filamentous just means like a ribbon. It's just like a long ribbon, like a whip, like a whip-like thing. And location, external, outside the cell, like you see here. It's not inside, it's outside. And then here's function. FX and function, movement, whatever, motility is the same thing. Okay, so some bacteria that are motile, this is how they move by, because they have that flagella there. And they move for different reasons. Maybe they're trying to locate a nutrient source, or they're trying to escape phagocytosis. You know, there's a reason for why they move, you know. So they have the ability to move. Composition, right? Composition is next. Composition, they're made of this protein that we call a flagellin protein. Okay. And some more information about flagellin proteins. These proteins, they're arranged like in these structures that we call filaments, and then several filaments make up a flagellum. Okay, that's kind of how they're arranged. And I think on the next slide, I think I kind of rewrote this a little bit for you, but again, if I'm going, I'll indent to the right. So it's like you have a flagellum, right? A flagellum, I'm telling you, is made of several filaments, so that's why I'm indenting to the right. Right? And then the filaments are made of flagellin proteins. Reminds me kind of a little bit about how muscle was arranged, right? Remember, like the muscle fiber, muscle cell was like the small. We had sarcomeres, and then we had the muscle fiber, and I forgot what the other ones were. Whatever. Okay, all these things. That's kind of how they are. Basically, a bunch of flagellin proteins make up a filament. A whole bunch of filaments make up the flagellum. It's kind of like what's happening here, right? So again, if I drew my entire flagellum, I'll just make it 3D thick for you. This is my flagellum, but the actual now filaments, I have several filaments in here maybe now that make up, I'll just draw a couple of them. I won't extend them all, but these are now my filaments. And then inside, what makes up the filament is my flagellin protein. So, I don't know, I'll put these purple little things here. These are now the flagellin proteins. Okay, so that's kind of like the arrangement of how these things work. Okay. These flagellin proteins, an example, this is a flagellin protein, the H antigen. example of a flagellum protein. Okay. And this is acting as a surface marker. What I mean by surface marker, it basically distinguishes between different types of cells, between different types of bacteria, depending on the type of H antigen present or the type of flagellum protein present. Okay? And this has to do with determining or distinguishing between the different strains / subspecies of bacteria, right? So remember this guy, the E. coli O157H7? You know it has letters and numbers, so you know it is a strain, right? It's not just regular E. coli in the colon, because that was called what? The E. coli in the colon? Normal microbiota, right? This is bad because it's a strain. I know it's a strain because it has letters and numbers. But this H7 is telling me that this H7, this is the type of, this is a flagellum protein. This is kind of like the H7 type of flagellum protein. Right? Because there's several types of flagellum protein. This happens to be the H7 type. I'll make something up. Maybe there's an H12, an H22, an H60, whatever. There's different flagellum proteins. This particular specimen has the H7 type of flagellum protein. This will distinguish it from another one that has whatever, H8 or H9. That's what I mean by helps to distinguish among the different bacterial strains, right? So, question for you. E. coli O157H7 is motile or not motile? Motile means movement, right? Does E. coli O157H7 move? Well, it has the H7, which represents a flagellum protein. If you have flagellum protein, you have filaments which make up a flagellum. What does a flagellum do? So the answer is yes, of course it does move. I know it does because the H7, the flagellum protein. You have flagellum protein, it's because you have flagella. So yes, it's capable of motility. Okay? So again, and various arrangements. This is what I was telling you the other day, I'm sorry, a couple minutes ago. You have various arrangements of flagella. Again, it can look like this, or it can have two of them. Or I told you it's kind of like a cluster of them here, like that. They just have different arrangements. You don't have to know the names, but there are names that are assigned to this based on the arrangement of the flagella. But I'm talking about the arrangement of the flagella, yes.
If bacteria has more flagella, would it mean that it will move a lot faster, or would it be?
That's a great question. If it has more flagella, does it move faster? Yeah, it should. Absolutely. You all have to work in unison, right? So, okay. All right. So again, this is basically what I just told you, but I think what I wrote on myself earlier was better. But again, here we can work our way backwards. The flagellum is made up of a bunch of filaments, which is made up of a bunch of flagellum proteins. And again, an example is something like H7. This is an example of a flagellum protein. Right? And H7 is a type of H antigen. Right? Like the surface marker, right? Again, this H antigen is found, you already know, we just talked about, in the pathogenic strain of E. coli called E. coli O157H7. Because that's basically what I just went over on the previous slide. Okay, any questions about the flagellum protein, how that's related to the H antigen or H7? H antigen, again, is just very broad. We can have different types of H antigen. H7 here, it can be H8, whatever. Okay? So, one more slide for flagellum, then we'll move on to the next structure. There's three general parts to the flagellum. Okay, we have the filament part here, which is the outermost part. That's the one we can kind of see that's visibly like moving. And there's a middle part here called the hook. This part here, right? And the hook basically joins the filament to the basal body. And the basal body is what anchors the actual flagellum, the actual structure itself, but the hook joins the filament to the basal body. Right, the basal body And again, the flagella, is this found in spirochetes or sporilla? Sporilla, right? Just to refresh your memory. Slide 12, okay, let's go back real quick. Dang it, what slide was that on? Oh, I thought it go with the, oh, here they are. So yes, we were talking about flagella, so yes, they're only found in sporilla. So guess what my next on our list is going to be? The, endoflagella is next. That is gonna be found, not in sporilla, but it's gonna be found now in spirochete. So let's go back now to slide 12. Or 13, I should say. Okay. So that's our next on our list. So now we're talking about spirochetes. Okay, the axial filament. You also know it better as endoflagella. It's the same thing, right? And again, this is not gonna be found outside. It's actually an intracellular structure. It's inside the cell. You won't really be able to see it, right? And here's an illustration, and this came from your book. It's kind of a lousy picture. I don't really like it because it's kind of deceiving because here's your endoflagella slash axial filament. It makes it look like it's on the outside, and it's not. I think what they really meant to do is to tell you that this is like a coronal section, right? Where they took out, they sliced it in half and removed the front half. This is the back half that you're seeing, right? That's why it's actually inside the cell. But it makes it look like it's on the outside, but it's not. Okay, because again, this is an internal structure. Okay, so again, endo means internal flagella. And the composition, also made out of proteins, but it's not important to know what the exact name is. For the flagellum, I told you it was a flagellum protein. People ask me, what's the name of this protein? I don't know. You don't need to know. It's just made out of proteins as well. Okay, so it's kind of analogous. And it is responsible for movement just like flagella, but the type of movement is very different. This doesn't really propel the specimen. It kind of gives it more of a rotational, like a corkscrew type of motion. It's not like it's gonna, travel very fast like that. This one kind of does more like a corkscrew type of rotational movement or spiral motion, right? And the reason kind of why it does that, this picture over here, which I drew myself, I'm very proud of this one. I did this one by myself. This is a cross section. If you were to slice an endoflagella, axial filament, what you'll have here, this is for example, here's the cell membrane in red here of your bacterium. And then remember, bacteria have cell walls. So within, like the space between the cell membrane and the cell wall, that's where you have the endoflagella. It's somewhere in there between the cell wall and the cell membrane. And there's several of them. Here I drew two of them, and they all kind of spin in the same direction. Here they happen to be both clockwise, so it's going to cause the green, the entire cell to spiral in one direction. So they all either go it either spirals in a clockwise direction or counterclockwise direction. Again, it just depends how that endoflagella is rotating. But again, that's where you find it. This is not an external structure. It's found actually inside the bacterium, between the cell wall and the cell membrane. Okay? Two more structures, and I put them all on one slide instead of doing two slides, but I guess I really should have put them on two slides. We have fimbriae, which is very different from pili. Right? You were asking about fimbriae. They're both external to the cell. This is one not inside, and they're both made out of proteins. It's not important to know the name of the protein, but they're made out of proteins and they're outside the cell. And the fimbriae, they're kind of more like what you have here. They're more like hair-like they're all over, little appendages. They're kind of smaller, a lot smaller than shorter, a lot shorter than flagella. And this also helps for attachment, the fimbriae. A lot of people always confuse fimbriae, that's for motility. It's not. Fimbriae is for attachment. Was flagella for attachment too? No. Flagella is for motility. These are, again, vice versa. These are not for motility. All right. Pili, for are involved in motility, actually, they have two functions. They're involved for motility, it's described more like as a twitching, so they really don't move that much, but the pellets does help them move a little bit. But anyhow, it's described as a twitching motility, and one of their main functions though is for DNA transfer. And when I say DNA transfer, I mean DNA from the donor, so it's a donor DNA, goes to the recipient, another neighboring cell. So the donor donates its DNA to the recipient cell. I have a slide on the next one that I drew out, right? And so for example, this blue one maybe is the donor cell, donor bacterium, and the green one is the recipient cell, recipient bacterium. Right, this blue DNA, the circular thing, this is the DNA. It's gonna donate some of its DNA to the green one. The green one, yes, has its own DNA too, but it's gonna donate some of it. It's actually gonna alter and change all the properties of the recipient cell by donating its DNA. Okay, and that's called this here. What's gonna be here in the middle to connect them, that's communicating, is the pilus, or they call it sometimes conjugation. Pilus. Conjugation has to do with sex. Bacteria don't have sex. There's no such thing as a male DNA transferring its DNA to the female DNA. They don't. But in some books they call it like quote unquote like bacterial sex, but they don't. Like bacteria are asexual, right? But I guess they were kind of getting cute with the name conjugate. You know what the word conjugation means? Have you ever heard I've never been to jail, but I mean like conjugal visit. Any idea what I'm talking about? Okay, it means sex. Okay, but bacteria do not have sex. There's no male and female. But technically speaking, when we talk about sex, it is transferring DNA. That's what the whole thing is about, the transfer of DNA. Right. Anyhow, wrong class, right? This is micro. So this is what I mean with the pilus, right? I have my donor cell that happens to have the green DNA, and it's gonna transfer some of its DNA through a pilus, right? To the recipient. And I told you the recipient already has its own DNA, the red one. And then what happens later with this recipient is gonna incorporate the green DNA, this is the green DNA right here, into it. So you agree the before cell looks very different from the after cell, because the after has this, which I should have put green, whatever. It has the green DNA in it. And it's now altered. This is now a very different cell now. Because there's genes in this piece of DNA. Maybe the green gene, the green DNA had genes for making a toxin. So before, this recipient made no toxin, and now guess what this recipient now is forced to do? Make a toxin. I can give you another example. Maybe this had genes for resistance to penicillin. This before was sensitive to penicillin, now what? It's not resistant. It's resistant now to it before. Mutation would be like if it's, mutation would mean like if this DNA changed itself on its own, right? Here, we haven't talked about this whole concept, but it's called, what is it called? Transformation, that's what it was. Like you transform a cell now into something that it wasn't before. We'll get to that later. And viruses can do this as well, and all this other stuff. But again, you're inserting DNA from somewhere else, so you'll never be the same as you were before. This red cell can never be the same. You altered it. You altered its DNA. Okay, we'll talk more about this later. You don't have to know this, but again, now this cell will split and divide into two, and all the future cells are going to have that green DNA in it. You completely change the rest of the population. Usually not for the better. Not for us, better for them, for survival. Okay.
More problematic.
For humans, yeah, it will be more problematic for us, because this is more immunology, because that one is like a war, us versus them. Okay? So now we're going to move on and talk about the cell wall. Next. Okay. And the cell wall, it depends. It's sometimes the outermost layer, but only if you don't have a capsule present. So we said the capsule was outside the cell wall, right? So it just depends. If there's no capsule, because I told you not all bacteria have capsules. If they don't have capsules, then the cell wall would be the outermost layer. Okay, so that has to do with location. Function-wise, this is the function. It prevents what we call osmotic lysis. Lysis is kind of like when something breaks up, right? Or bursts, right? So it kind of protects the cell from bursting. Is that the right word? Bursting? Yeah, to burst, right? It protects the cell, obviously the cell membrane, but it protects the entire cell. Composition. And we've talked about this thing called peptidoglycan. So this is the composition. It's made out of that peptidoglycan, which is basically, again, break up the words, peptido and glycan. So it's made out of a sugar that's connected to a protein, found again where? In the cell wall. And I think we've talked about this a little bit in the first chapter already, right? And we'll talk about this a lot in detail next week in lab, about peptidoglycan, because our focus is going to be on the cell wall next week. And you're going to stain the cell wall with bacteria and look at them. You're going to determine the differences in the cell wall based on colors, okay? Which I'm going to talk about here as well. So a major ingredient in the cell wall of bacteria is this thing called peptidoglycan. Not all cells are uniform. You have some bacteria that have a lot of peptidoglycan, and you have some cells that have very little peptidoglycan. So this amount of peptidoglycan varies. Amount, AMT, amount in the cell wall varies. Okay, sorry for all the abbreviations, but you have to kind of get used to writing abbreviations when you take notes, right? Amount of peptidoglycan in the cell wall varies. You can have a lot versus a little. If there's a lot of peptidoglycan in the cell wall, then these are called gram-positive bacteria. If there's very little peptidoglycan, then it's gram-negative bacteria. Okay? So we're looking at bacterial cell wall differences, right? And we'll talk more about this in lab. Again, when I ask people questions in lab, because sometimes I used to do a verbal quiz that was entirely verbal, but that takes too long. I don't have time. I have 3 sections this semester. It was like a job interview, right? Like it took 5. It depends. Some people are really good. I was done with them in 3 minutes, and other people, this was like a 20-minute. I was like, oh my god, it was just like, yeah, it was taking a long time. It was easy. Everyone would get 100 because I would like clue you in if you were giving me the wrong answer. Anyhow, we don't have time for that, unfortunately. But why am I telling you? Oh, because people would tell me, oh, we have gram-positive bacteria versus gram-negative bacteria. I'm like, okay, so what does that mean? How are they different? One is positive, one is negative. Okay, nice try. You see why it took 20 minutes sometimes. Okay. Again, we're talking about cell wall differences, right? How are the cell walls different? One has a lot of peptidoglycan, the other cell wall has very little peptidoglycan. So it's all about cell wall differences. It's not like, oh, they're different, because one's positive, one's negative, and so forth. The colors also are important, but that's not the main thing. Gram-positive are purple / violet color. You know what violet, like the flower of violet is purple. That's another thing I'd ask people. They're like, what color is gram-positive? I don't know, okay. What dye did you use? There's a dye I'm talking about, it's called crystal violet that we used to stain. I go, does that give you crystal violet? No. Violet, what color is violet? I don't know, okay, it's like, it's purple, so they stain purple, right? Okay, because of the dye that I'm about to talk on the next slide. And the gram-negative, I put them red because they stain like a reddish pink. Stain reddish pink color. And you'll get to do this next week in lab when we start dyeing things. So again, this one will stain purple, violet, this one will stain reddish pink. This same slide, I'll have it on a PowerPoint next week also for the lab PowerPoint, but this is the lecture PowerPoint. Again, showing you the differences between gram-negative and gram-positive bacteria, right? So just to complete the thought, bacterial cell wall is what I'm talking about. Gram-positive bacterial cell wall. Because again, when people tell me gram-positive, gram-negative bacteria, they don't understand it. I ask them, what's the difference though? What about the bacteria? I don't know. No, yes you do know. The difference is in their cell walls. That's what you mean when you say gram-positive bacteria, gram-negative. Their cell walls are different. Again, by the amount of peptidoglycan, right? Didn't we say that gram-positive you had a lot? The purple is the gram-positive. The purple is the peptidoglycan. You you have a very thick layer of peptidoglycan, you have a lot of peptidoglycan. Gram-negative, look, it only has this one single layer. So you can see that the amount of peptidoglycan varies from a lot for the gram-positive to very little for the gram-negative, right? This whole area here, by the way, this is all the cell wall. I put CW cell wall. This is not the cell wall, this is the cell membrane. Right? Remember, the cell wall is outside of the cell membrane. Okay? Gram-negative are interesting because they have a lot more things going on. Gram-negative have this outer membrane, and external to the outer membrane, you have this other very important compound that's called lipopolysaccharide. We know it better as LPS. It's okay to just call it LPS. Change color. LPS, lipopolysaccharide. Lipo is referring to lipid, so this is a sugar that has some sort of, again, polysaccharide is a sugar. It is a sugar that has a fat or a lipid attached to it. Only found exclusively in gram-negative bacteria. LPS is not present in gram-positive, right? And I'm going to talk a little bit, well, I don't know about today, but I have some more information, a little bit of coming up about LPS, like in 2 slides. A little bit more detail about LPS. You know it has lipid in there, and it's found in gram-negative cells. Cell walls, gram-negative cell walls of bacteria. Okay. Before I move on to the next slide, I just remembered also very important. When students see these words gram-negative and gram-positive, and they focus on the negative and positive, this has nothing to do with charges. Don't get that confused. I would have people that tell me, like, when I ask them what's the difference, they're like, one is positive, one is negative, which is not the great answer, but they're like, start telling me about charges. Well, the gram-positive, it has like a positive charge, and it attracts. And you're just like, yeah, that's not a good conversation. It's going nowhere, right? It has nothing to do with charge. I don't know why they named them this way. No one asked me. If they would have asked me, I would have called this one. I would have called this one the gram. I would have called this gram little, like gram less, like not gram less. What am I talking? It has to do with peptidoglycan. Like you don't have much, like gram a little peptidoglycan versus gram a lot peptidoglycan. It has to do with the amount of peptidoglycan. Right? Because usually when we look at positive and negative values, like you get a test result, when it says negative, that means nothing is there, right? Well, of course, nothing is there, you have very little. Versus if the test result is positive, something is there, right? The something that I'm talking about is what? Peptidoglycan. But don't get confused with charge. It has nothing to do with charge right here. So people are like, yeah, they try to explain that the gram positive attracts the gram negative cell. Some more differences. You already know this because I already talked about this, right? The 2 differences. One has a lot, it's a thick peptidoglycan layer, and a thin peptidoglycan layer. There's this other thing called teichoic acid. You don't have to know much about it except that gram-positive has teichoic acid. I'm not going to test you on this, but I mean, teichoic acid basically, it provides rigidity and strength to the cell wall. It's present in the cell wall. And it attracts ions to it. If you have a lot of ions in the cell wall, it makes it kind of stronger cell wall. Okay, but so gram-positive have teichoic acid, gram-negatives do not. How I always remember it when I think of gram-positive, right? I look at my positive, right? Like the positive. Doesn't a T look like this? That's how I remember it. So this is not a T. This is gram-negative. This is not a T. So I know it doesn't have teichoic acid. T is this one. So gram-positive, yes, they have teichoic acid. No outer membrane, but we already said that the gram-negative does have an outer membrane, and there's even an LPS attached to it. Here, let's go back. It's right here. Right? Outer membrane, that's gram-negative, and it has the LPS, these little green, look like little trees, right? Like conifers, Christmas trees on top of it. Again, LPS and outer membrane, that's only for gram-negative bacteria. Gram-positive don't have that. Okay. In terms of medication, again, you already know this one about medication, penicillin. By the way, what scientists discovered this accidentally? Who? Fleming. Alexander Fleming, right? Okay. Where did this come from? From the fungal mold, right? So it came from a mold, right? You don't have to know the genus name, but it was penicillium. So yeah, this is an antibiotic, right? And when we say sensitive, what we're trying to say here basically is that penicillin kills or destroys gram-positive bacteria. So if you are infected with a gram-positive bacteria, it's okay to be given penicillin, right? The opposite over here, when we say that gram-negative bacteria are resistant to penicillin, then we're saying that penicillin will not kill gram-negative bacteria. That's what resistant means, that this bacteria will not die. It's resistant to the penicillin, right? So it will not kill gram-negative though. Gram-positive, yes, it will. Okay, so we have a chapter at the end of the semester that is a drug chapter that we'll cover. We'll talk more in detail about how penicillin exactly works. You don't have to know this, don't write this down, but penicillin basically, it inhibits the synthesis of peptidoglycan. Now peptidoglycan is very difficult to have a strong cell wall. Okay, most bacteria need peptidoglycan. Right, we'll save that for exam number 4. Okay. All right, next slide. Let's focus now on gram-negative. We know they have an outer membrane that's not found in gram-positive. Remember, we have also LPS attached to the outer membrane. But yeah, this outer membrane is a major component of the gram-negative cell wall. Because remember, outer membrane is part of the cell wall. Right, I just told you this whole thing here, starting from here all the way up to here, this is the cell wall. Right? Yes, it contains peptidoglycan, it contains an outer membrane, and then it contains the LPS, lipopolysaccharide. So let's talk about what are the things that make up LPS, the lipopolysaccharide. Oh shoot, I passed it up. Okay? Yes, it contains lipids and carbohydrates, right? The lipid, lipo, right? Lipopolysaccharide, 3 components. And I always know it's 3 components because there's 3 letters in LPS. L for lipid, and it's called the lipid A component. So this whole giant green thing is supposed to represent the LPS structure for gram-negative, because again, we're talking about gram-negative cell. This is a gram-negative cell. So again, you can tell because it has an outer membrane and an LPS, so you know this is gram-negative, not gram-positive, right? So this part here, you don't have to know that it's anchoring in the outer membrane, whatever, but here's the lipid A portion of it, and your other 2 portions, they're sugar portions called the core and the O part, O polysaccharide and core polysaccharide. Here's the core, and I guess the O part sticks out, right? So it's kind of the order here, the sticks out. Right, and the core is kind of like the middle portion. Okay. The lipid A portion, you know, which is embedded further inside the outer membrane, it actually functions as a toxin. Right? Some bacteria produce toxins, some do not. Those that produce toxins, you want to take a wild guess, are they gram-positive or gram-negative? Those bacteria that produce toxins, they're gram-negative, because only gram-negative have LPS. The L part in LPS is the lipid A part that acts as a what? Toxin. Okay. So yeah, usually bacteria that give you that are responsible for gram-negative bacteria type of infections, those usually worse than gram-positive. You have more side effects. Okay, but I'll save that when we study the diseases in exam 3. Okay, and then I told you these other 2 polysaccharides, I told you this was the middle one. It just joins the lipid A to the O polysaccharide, this core. And then the O, this is like a surface marker because it's the one that sticks out, right? So this is acting as an antigen or a surface marker. It just helps distinguish between the different gram-negative bacteria, right? Based on the different type of O polysaccharide. Because gram-negative, they all have LPS. You have to understand, you have different bacteria that are also gram-negative. They don't all have the same LPS. Their O part differs. So that's acting as a surface marker, as an antigen, to distinguish between the different gram-negatives. All right? Still talking about the cell wall, and we'll cover this a lot more in lab, because in lab you actually perform the actual mechanism for staining a cell, determining whether that bacteria that you're looking at is gram-negative or gram-positive. And yes, you can tell by the color, for sure. All right, but you have to know a little bit more about that. But again, we do the staining, and it's called gram staining. Again, I think I have the same slide on the next week lab PowerPoint. Probably developed by some doctor named Dr. Graham. They named it after him, gram staining. And again, this is to distinguish between two types of cell walls: gram-positive cell wall versus gram-negative cell walls. This staining procedure. We get to do this next week, not this week, but next week in lab from the specimen that you got from home. That you'll bring back over. Don't forget to bring your send me an email. Bring back your Ziploc bags. All right. But so this is what you kind of do, and here are the steps. There's 4 steps involved here, and I'll go through these more in detail. But 2 of these steps involve a dye. Crystal violet is a dye or a stain, and so is saffronin. It's like a red-pink dye. Okay. So what they do here, and you have to kind of go in order. So you use crystal violet first, right? That's number 1, and then at the end, number 4, you use the saffronin red dye. Again. You start off with crystal violet, which is purple. Violet we established was purple. So this is your visual field. You're looking at the microscope. So, and this is a mixed culture probably, because I have cocci and I have bacilli. So if I'm using crystal violet, everybody stains violet, correct? Whatever you do the second step. Anyhow, the third step, you use this alcohol, and I'll talk more about that in detail. Okay, but what you notice at the end of the alcohol use, what happened? Alcohol decolorizes. What's a decolorizer mean to you? It does what? Removes the color. Decolorizes removes the color. Alcohol removed the color for everybody? No, it didn't. It only removed the color for the bacilli. Remove the color for bacilli only. The rod shape. It did not remove the color from the cocci. It's still purple, right? And then you go to the step 4, you use the saffronin red color, and look who stained red, the ones that were colorless. So this is colorless. If it's like this, clear, this is colorless. Because of alcohol. So you understand why the rods were able to pick up the red color? Because previously you decolorized them. So yes, they can pick up the red color. The purple did not pick up the red color because there was a color already on it. You cannot superimpose another color on top. If you wanted the cocci, the spheres, to stain red, what did you have to do? What would you have to have done earlier? Remove the color. But it did not remove the color. The alcohol did not. They remained purple, so even though you added red on top of it, in the last step, it did not pick up the red color. Because again, you have to remove a color first before you expect that one to pick up the second stain. Okay. Why it didn't do that, that's the story for lab. Okay, you're gonna have to wait and suspect. But there's a reason why they did not get decolorized the concept. So that's why at the end, it looks like this. Okay. You have a mixture, some gram-positive, some gram-negative. Okay, just give me like 1 minute, okay? Just let me finish, I think, this slide. All right. And this is what they would look like, right? So you can tell, this is purple, so this is gram-positive, here gram-negative. Okay, so for identification purposes. And again, the gram staining tells us a lot of information about shape and guess what else? Arrangement. What shapes are the gram-positive? Are they rod or round? Rod, so these are bacilli. I can tell thanks to staining. I would never find this information out if I didn't stain. Here, these are also bacilli. People are like, oh, well, trust me, they're bacilli. They're just very short bacilli, right? What's the arrangement of something like this? Streptobacilli. So you see how you can tell shape and arrangement from the staining procedure. See, that's the whole point why we want to stain. And we can do other things, we can measure, we can do a lot of things, right? But this is more the discussion for lab. So this will overlap nicely with next week's lab. So I will continue next week's, this is the last slide for the, I won't do it today because there's another class coming in here. And then, like I said, on lab, I just have to take some time. Okay.